Okay, so let's start with our first cell adaptation, that is hypertrophy. See, hyper means more, and trophy means size. When we talk about size, we use the word trophy. Okay, let's say these are normal cells, and after certain stress, they will change their size and appear like this. So I will call them hypertrophy cell. Okay, this is one of the cell adaptation. Uh, let let me give you a concept here. Cells can be dividing. Cells can be non-dividing cells. Okay. The example of dividing cells are germ cells, stem cells. The example of non-dividing cells are basically any somatic cell which do not want to get divided, because somatic cell can divide and will not also have choice of not dividing, because a somatic cell has got a Hayflick limit. We will learn that later in our discussions. So let's say non-dividing cell. See, any kind of stress will come. Let's say any stress is coming here. Dividing cell have two adaptation to do. First is hyperplasia. It can do hyperplasia. The next is hypertrophy. But a non-dividing cell cannot divide. And for the from this example, it cannot do division. So there will be no hyperplasia. There will be only hypertrophy. See, hypertrophy non-dividing cells are exclusive for hypertrophy. Do you get what I am trying to say? See, non-dividing cells can adapt only by getting hypertrophy. They cannot do hyperplasia. So this is a very good concept. It can be checked by in your Viva or in any competitive exams. So let us discuss how the hypertrophy occurs. Okay, what is the mechanism of hypertrophy? See, I would I would take an example of heart muscles. Let's say I am saying here non-dividing cells. Na, these are basically heart muscles or any kind of skeletal muscle. These two tissues are the normal ones which are discussed in hypertrophy. Let's say what is the first mechanism? Any mechanism. Okay, first mechanism is actually protein. When there is a increase in protein synthesis inside the cell size will increase, because for cell size to increase, its structure will need to increase, and the structure of any cell is formed by the proteins. So if the overall protein synthesis increases, cell size will increase, and we can say the cell got hypertrophy. Let us discuss how this actually occurs. There are many sensors here on the cell, which are mechanical sensors, which can sense mechanical stress, or there are few growth factors receptors. Growth factors can reach to cell, okay, in as a feedback form or as a anticipation move. Some vasoactive factors, I will give you the example, like for growth factor it can be TGF beta, tumor growth factor beta, or IGF-1, which is insulin like growth factor 1. These two growth factors, as examples for vasoactive factors, it can be vasoactive means they will stimulate vessels for vasoconstriction. Okay, so it can be alpha agonist, it can be endothelin 1, or it can be angiotensin 2. You will read all these receptors, what are their drugs associated with them in pharmacology. As for the namesake, I have given you three examples. All these three, okay, these three will stimulate the cell and gives a signal, okay, And there is a thing called signal transduction pathway. They all three sensors will give a signal to the cell, give a signal transduction pathway via the transduction pathway. Okay, what are the signal transduction pathway in here is, say if the stress or the condition is physiological, if the cell there is a physiological state of stress, or if it is a pathological state. In physiological, the signal transduction pathway used is, the name of that is PI3K AKT. Full form is phosphoinositol, oh sorry, phosphoionoside 3 kinase. I will write it. Phosphoinositide 3 kinase. Okay, and as if the stimulus is pathological and it is pathological hypertrophy, then the receptor will work is GPCR, signal transduction pathway, for pathological hypertrophy, and it is for physiological hypertrophy. Both these sensors, okay, both these signal transduction pathway will stimulate few factors, namely GATA GATA for NFAT, neutrophil factor activating, and MEF2. All these three are actually transcription signals. Okay, what phosphoinositide 3 kinase and GPCR does is they stimulate the transcription signals, namely GATA4, NFAT, and MEF2. Transcription is a process of synthesis of proteins, and if the signal for transcription is stimulated, it will lead to protein synthesis, and protein synthesis will lead to cell size increase. This is the mechanism of hypertrophy, how the hypertrophy will occur. The next thing, next point you need to know about hypertrophy is, okay, I will erase this. That is the type of protein change. Okay, what is the type of protein change occurring when the cell is hypertrophied? Let's take an example of cardiac muscle. I am saying, hey, there is a cardiac muscle. This is our heart. A patient comes. Okay, a patient comes and he has hemodynamic instability in his chest, in his body. Overall, in simple terms, he is a patient of BP. Okay, he cannot control his BP. So that in that person, the first thing will be heart will try to adapt, and how the heart adapts is by hypertrophy, and the heart will undergo this pathway, this whole pathway, and the cell will become hypertrophied. What other things will change here? Okay, the one other thing is in hypertrophy, heart, as a heart as example, in hypertrophy, what happens is there is a change in type of protein. What I'm trying to say is, what I'm trying to say is that if there is a alpha myosin unit in the muscle, it is a alpha myosin unit, it will be changed. It will be replaced by beta isoform. Okay. So you will say, oh, what is the difference? Okay, why it is changing? The reason is, see, alpha myosin act as a fast contractor. It will contract the muscle faster, and but the beta isoform is for slower contraction. It will, if the change is that the fast contracting heart muscle will be replaced by slow contracting protein. Okay, and these slower kind of beta isoform are usually found in neonates. It's a point that you need to remember is that in neonate the heart muscles contract smoothly and slowly. It is not, oh, okay. Sorry, muscles contracted faster. Heart rate is more, but the muscle type is very slower. Okay, See, the changes in hypertrophy in heart is if there is adult protein, it will become like child and neonatal. It will start resembling a neonatal heart if there is hypertrophic changes in heart. The third point you need to remember is there are genetic changes occurring in hypertrophied cell. How do I explain it? The saying, if this is a cell and it undergoes hypertrophy, then there will appear few receptor here, okay, which are actually embryogenic in nature. I am saying any hypertrophied cell resembles neonate form. Okay, we've seen the previous point that alpha myosin will change to beta myosin. Overall, the point is adult cells are reverting back to their embryonic regions, and adult when they are doing so, few embryonic receptor will appear in them. One of that will be ANP, ANF. Sorry, ANP. My mistake. ANF, atrial natriuretic factor. What I am saying is atrial natriuretic factor. What does it mean? Okay, the atrial means I'm not saying it is only found in atria, it is found in both atria and ventricle of heart. The next thing is natri. Natri means sodium. Uretric means urine. Sodium loss in urine. And it is a factor which causes sodium loss in urine found in atria and ventricle both. What will be its function is ANP, it is actually peptide. It will cause sodium loss, which will inherited loss, H2O loss or water loss, which will decrease the volume and decrease the workload of heart. So all these 3 points which I am discussing here, first is the mechanism of hypertrophy and other 2 points in cardiac hypertrophy changes for adaptation. The first is the change in type of proteins. The second is the formation of the occurrence of atrial natriuretic factors, which will all of these are for decreasing the workload of the heart. Okay, thank you. This is all the hypertrophy we need to discuss. Okay, we will see you in next topic.